Thank you all for joining us. You've logged into Grand Rounds presented by the Department of Psychiatry and Behavioral Sciences. Welcome. We've got a great presentation for you today. I do want to give people a few moments to get logged in. Usually takes 1 or 2 minutes for everybody to get on. So I will introduce myself and go over a few of the logistics of our Grand Rounds series before we begin. So hello, my name is Jed Thompson. I am the Associate Director of Communications for the department, and I'm also the host of our Grand Rounds series. So a few logistics to cover, and then we'll get started. First, you probably heard when you logged on that we are recording this session. So we will have this posted in our Grand Rounds archive sometime next week. If you would like to check that out again, or if you'd like to check out any of our Grand Rounds going back for about 10 years now. It's a pretty big archive, so take a look at that. We have a survey that will show up at the end of the presentation. So if you'd like to give us any feedback about this particular Grand Rounds or just our Grand Rounds series in general, once the webinar closes, you will see a link for that survey. So take a couple minutes to let us know what you think. In terms of Q&A, so we've got the Q&A feature and we've got that chat feature going today in the webinar. We prefer to use the Q&A feature, so if you have any questions, go ahead and type those in. We're going to try to save about 5 minutes or so at the end for questions, so get those queued up. I will be disappearing during the talk, but then I will come back at the end and we'll go through those questions. And if you like to plan ahead, next Friday, October 24th, we'll be joined by two people from our department, Dr. Benjamin Danielson and Dr. Eric Truppen, and they will be talking about youth incarceration. So join us for that next week. And why don't we go to the next slide now? Thank you. So now I have the great honor of introducing today's presenter, Dr. Jennifer Forsyth. Dr. Forsyth is an assistant professor in the University of Washington Department of Psychology and an affiliate faculty member with the Institute for Public Health Genetics. She is also the director of the Genetic Risk and Neurodevelopment Lab, also known as the GRAND Lab, where her research seeks to understand how people develop serious mental illnesses, such as schizophrenia, with an emphasis on understanding the pathways from genetic variation to neural and systems level changes. So in 2024, Dr. Forsyth became an associate member of the American College of Neuropsychopharmacology, and she just recently received the UW DeVita Teller Distinguished Faculty Award in Psychology. So with that, welcome, Dr. Forsyth. Thank you for being here, and I will turn it over to you now.
Fabulous. Thank you so much, Jed, for the introduction, and thank you so much for the invitation, for having me here, and everyone who's attending. I'm excited to get to share some work that we've been doing in my lab in recent years. So the title of this talk is Advances in Understanding Schizophrenia Using Genetic and Developmental Frameworks. Okay, so what do we know about schizophrenia? So it's a debilitating neuropsychiatric disorder. And from a clinical perspective, it's been largely defined by the presence of psychotic symptoms like hallucinations or delusions that typically emerge in late adolescence or early adulthood. And really, despite decades of a lot of research, there's still a lot that we don't understand about the core biological processes that underlie the disorder. And our current treatment strategies, which generally involve the introduction of treatment after the onset of full-blown psychosis, typically antipsychotics, this kind of treatment strategy generally isn't effective enough to prevent the decline in functioning and the disability that affects so many patients. So, for example, rates of unemployment and social disability are really high. Patients overall have a reduced lifespan by 10 to 20 years on average, and part of that is due to high rates of suicide that are highest in the first few years after symptom onset. So there's just a lot of personal and broader societal suffering associated with this disorder. And so overall, this tells us that there's an urgent need for us to do more and really try to find new ways to improve patient outcomes and understand its etiology. So what are some key facts that I think we need to think about if we're trying to understand schizophrenia? First, it's a broad syndrome. So while we've typically focused on psychotic symptoms of the disorder, it's also associated with various sensory, motor, and cognitive signs and symptoms, many of which emerge prior to illness onset. So for a long time, this has suggested that schizophrenia is best thought of as a neurodevelopmental disorder. The cognitive impairment in schizophrenia is now generally considered a core component of the disorder because it's present prior to psychotic symptom onset. It's one of the most stable features of the illness and is also a strong predictor of long-term outcome. And from neuroimaging and postmortem brain studies, we know that there are disruptions within and across many regions and neural circuits of the brain. And we also have known for a long time from family studies that schizophrenia is highly heritable, around 80%. So that's also told us for a long time that genes play a major role. We're also increasingly learning that many risk factors for schizophrenia, both genetic and environmental, are shared with other disorders. And patients differ in the specific risk factors that they carry or that are predisposing them to developing schizophrenia. So to me, these have always seemed like a sort of set of puzzle pieces that we need to make sense of and try to put together if we really want to understand what are the mechanisms that lead to this clinical syndrome, including how those mechanisms differ from other disorders and between patients, and how we can best improve patient outcomes, including ideally through early prediction of the disorder and earlier intervention. So for today, I'm going to tell you about some work that we've done trying to leverage recent gains in genetics to understand the path from genetic risk to complex syndrome in schizophrenia from a brain developmental perspective. And then I'm going to tell you about our work trying to understand some of the clinical and genetic heterogeneity between patients. And finally, I'll tell you about some of our ongoing work trying to move us towards earlier prediction of schizophrenia. Okay, so with the rise of large-scale genetic studies, we've really learned a ton about the genetic architecture of schizophrenia over the last decade and a half, including that it's complex and highly polygenic. So it involves many common single nucleotide variants that individually each have a small effect. That's what you can see from the odds ratios associated with the turquoise dots down in the lower right part of this plot, where you can see the allele frequency in a population on the x-axis and then the relative risk for schizophrenia on the y-axis. So those little turquoise dots for common variants, you know, the effect sizes are really tiny. Risk also comes from recurrent copy number variants, or CNVs, where large stretches of DNA that often span many genes are deleted or duplicated. And in the case of a deletion, this usually means that there's going to be a big decrease in the expression of the proteins that are encoded by the deleted genes. Recurrent CNVs associated with schizophrenia are rare in the general population, but when they are present, in a given individual, they can really increase risk quite substantially, which is what you can see from the much higher odds ratios associated with the blue dots towards the left of the plot. And then aside from deletions, other variants that have protein truncating or loss of function effects also increase risk for schizophrenia. And some specific genes have been implicated by these other kinds of protein truncating variants. I mean, that's what you can see from the red and yellow dots for some specific genes also towards the left of the plot. Okay, so we've also learned in recent years that risk variants for other disorders, especially earlier onset neurodevelopmental disorders like autism and intellectual disability, sometimes overlap schizophrenia. So here on the left, you're seeing the parallel plot for variants associated with autism. And you can see that, for example, some of the specific recurrent copy number variants in blue on the left for autism and on the right for schizophrenia, they're actually associated. It's the same specific copy number variants that are associated with both disorders. So for example, someone with a 22q deletion, which deletes about 45 protein coding genes, has a strongly increased risk for schizophrenia, but also for autism. And though it's not shown here on this plot, actually also for intellectual disability and ADHD, for example, as well. So some variants really have what we call pleiotropic effects. So these kinds of recent findings in genetics allow us to ask on a systematic genome-wide basis, do risk variants for schizophrenia converge onto specific neurobiological processes? And if so, to what extent does this occur across common and rare variants or different classes of variants? We can also ask the same kinds of questions for earlier onset disorders like autism and intellectual disability. What neurodevelopmental processes do they seem to converge on? And are the associated neurodevelopmental processes similar or distinct from schizophrenia? So to answer these questions, we took advantage of the fact that the selective expression of genes in a given cell at a given time really is what determines the function and the identity of that cell. This selective expression is what drives the development of the brain through stages of early proliferation of cells, differentiation of cells into different cell types, like this neuron or the oligodendrocyte in this little figure, during fetal development, the initial formation of synapses, and later the refinement of synapses into the mature circuits that characterize an adult brain. So what this means is that genes that work together to drive different neurodevelopmental processes or to give cells stable identities, they tend to have similar patterns of expression. And especially when it comes to brain development, where gene expression changes really dynamically to drive sort of the building and refinement of the brain, that means that data-driven clustering techniques like weighted gene co-expression network analysis or WGCNA can be a really powerful strategy for organizing genes into the developmental cellular and molecular systems that they operate in. So this is from some work that we did several years ago. We leveraged data on genome-wide gene expression, so transcriptomic data, spanning over 17,000 genes from over 1,000 samples using the publicly available BrainSpan transcriptomic set, where those 1,000 BrainSpan samples were taken from 16 different regions of the brain, and they spanned late embryonic development. So starting at 6 weeks post-conception, we kept the samples that went until 30 years of age. So basically, we were trying to capture gene expression changes across the full span of brain development. And so when we applied weighted gene co-expression network analysis to that set of samples, this organized the 17,000 or so genes into 18 co-expression modules that we could then validate using independent data sets. And we found that these 18 modules were enriched for biological processes associated with distinct aspects of neurodevelopment. They were also enriched for transcriptional co-regulation and for known protein-protein interactions. And while I won't go into all the details of these modules, when we mapped their expression trajectories over development and looked at what the genes in these modules were associated with, we saw that the first half of the module, so M1 through M9, they had their peak expression during fetal development. And they mapped largely onto early brain developmental processes, so like DNA replication, embryonic cell division, neuronal differentiation. Whereas the latter half of the modules peaked in expression during postnatal life and were involved in later aspects of brain development, like the M10 module, which was involved in myelination, or the M15 module that was involved in postnatal synaptic signaling and plasticity. So we could then take those modules and ask which of these modules are associated with genetic risk variants for schizophrenia or for autism or intellectual disability. So we compiled summary statistics and gene lists from the largest genome-wide studies for different classes of risk variants for these disorders in the literature at the time. And we asked which modules were, the modules ranged in size from about 50 genes to nearly 3,000 genes, which those modules are enriched for risk variants for these disorders. Okay, so what did we find? Looking at this plot, we've got the 18 neurodevelopmental modules along the x-axis, and then the different variant data sets along the y-axis for schizophrenia, autism, and intellectual disability. We saw that there were 3 modules that were significantly enriched for risk variants for schizophrenia. M15 showed the strongest signal. It was enriched for both common variants and copy number variants. associated with schizophrenia. M13 was also enriched for common variants, and M7 was also enriched for common variants with some enrichment showing also for rare variants as well. We also saw that the same modules that came up for schizophrenia, they showed some enrichment for autism and intellectual disability risk variants, but there were 4 additional modules that were more distinctly associated with autism and intellectual disability, some of which showed really strong enrichment signal like M1. So you can see the 4 modules were M1, M4, M6, and M12. Okay, so what are these specific modules doing then? Highlighting a couple here that were strongly enriched for schizophrenia risk variants, but also showed some enrichment for autism and intellectual disability. M15 is a module that increased in its expression across development. That's what you can see from that sort of like line plot, where you've got age 6 weeks post-conception through 30 years along the x-axis, and then on the y-axis, you've got the level of expression. And then you can see from the right, that's showing the gene ontology terms that were significantly enriched for genes in the module. So this is a module that was involved in synaptic signaling, refinement of neurotransmission, and learning and memory. And then risk for schizophrenia, but also autism and intellectual disability was also enriched for M13, which also increased its expression across fetal development, but then plateaued around birth. And genes in this module were involved in more basic aspects of synaptic transmission, like ion channel activity and regulation of membrane potential. So regulating sort of overall neuronal excitability is how we thought about this module. Okay, so what about some of the modules that showed more specific enrichment for autism and intellectual disability? Highlighting a couple of them. M4, you can see it peaked in its expression during mid-fetal development. And it was involved in neuronal differentiation and morphogenesis, or the outgrowth of axons and dendrites that form synapses. And then M1, down at the bottom, showed its highest expression during early to mid-fetal development, with its genes involved in regulating transcription and chromatin organization. So this module appears to be involved in orchestrating gene expression changes that are really critical for driving early brain formation. And indeed, actually the most connected, what we call hub genes in this M1 module included SOX11 and SOX4, which are transcription factors that are known to promote neuronal differentiation from embryonic stem cells. So it's suggesting that actually there's probably a link between these 2 modules, where maybe a lot of the genes in M1 are actually, genes that shape transcriptional changes that drive things like neuronal differentiation and morphogenesis. Okay, so coming back to the model, what did we learn about the neurodevelopmental mechanisms underlying schizophrenia versus earlier onset disorders? It looks like one primary path from genes to complex syndrome in schizophrenia may be altered neuronal excitability and synaptic signaling, which then can ultimately disrupt the activity-dependent refinement of synapses and circuits during postnatal development, which is a time when environmental input is especially critical for shaping the maturation of neuronal circuits. And this could therefore lead to the emergence of the broad schizophrenia syndrome across development. Whereas it looks like genes that maybe lead to earlier onset neurodevelopmental disorders like autism and intellectual disability, they seem to be additionally impacting and strongly impacting earlier brain processes like neuronal differentiation and morphogenesis, which would lead to greater disruption in the initial formation of neuronal circuits during prenatal development, as well as disrupted circuit refinement postnatally. Okay. Now, so that's thinking about risk variants and how they might impact brain development when we think about the clinical population level, but what about heterogeneity, right? So like I said, the specific genetic variants that confer risk for schizophrenia, we know that they actually differ from one patient to the next. For example, we might have one person with schizophrenia where their genetic risk loading is largely coming from common common variants, whereas another might have a copy number deletion that is driving the majority of their risk. We also know that there's a lot of clinical heterogeneity between patients. So while cognitive functioning is, like I said, an important predictor of long-term outcome, and on average is about 1 standard deviation lower in patients compared to controls, there can also be a lot of variability between patients, with some patients having average functioning or above average functioning, and then others functioning in the borderline or intellectual disability range. And similarly, while late adolescence or early adulthood is the most common age of psychosis symptom onset, some individuals have their first psychotic symptoms as early as in childhood, which is a more severe phenotype associated with poorer outcomes. So this raises the question of whether different clinical presentations or more severe phenotypes in schizophrenia are associated with differences in the underlying genetic architecture between patients, or possibly differences in the loading of genetic risk variants onto different biological processes. So to start to answer these questions, we've been working in a cohort of about 1,500 individuals where we have clinical and cognitive phenotyping, including 615 mostly recent onset schizophrenia spectrum individuals, as we also have data on 234 relatives of psychosis patients and 583 controls. This is data that my collaborators had collected across studies across many years, and we've now pooled and harmonized the data to do these analyses. And a subset of this cohort also have neuroimaging data. And then in terms of the severe phenotypes in this cohort, about 20% of the patients had cognitive functioning in the borderline intellectual functioning range, so where their estimated IQ is 85 or lower, and 6.5% had psychosis onset in childhood, so by age 12. And in this cohort, refining our research question a little bit, we've started with CNVs. And so we first asked whether severe phenotypes like child onset psychosis or borderline intellectual functioning are associated with the occurrence of those recurrent CNVs that I mentioned earlier, or with differences in the number of genes that are deleted by CNVs, where the genes might be involved in different aspects of brain development. And you might ask, okay, why are we focusing on rare copy number variants to start? Like I mentioned before, we do now know that certain recurrent CNVs are among the most highly penetrant risk factors for schizophrenia. They're also associated with earlier onset disorders like autism and intellectual disability. We can also call CNVs pretty easily from genotyping data, and risk scores based on CNVs are less subject to issues like population stratification than polygenic risk scores. So pragmatically, this felt like a reasonable place to start in this cohort. And from a mechanistic perspective, the deletion of a gene typically has a much larger effect on the expression of the protein than common SNPs do. So there's a good reason to think that copy number variants could drive these more severe phenotypes. So in this cohort, we called rare CNVs using standard processing pipelines, and then we annotated our CNV calls for overlap with known risk CNVs that have been previously associated with schizophrenia or associated more broadly with any neurodevelopmental disorder. So including schizophrenia, autism, intellectual disability, other developmental disorders, which from here will be called our broad NDD risk CNVs. And then we also asked whether the loading of rare deletions affecting genes in the 18 neurodevelopmental gene sets that I described before was associated with these severe phenotypes, as well as some global burden CNV scores. In particular, the total number of genes that somebody carries that are deleted by rare CNVs, and the total sum of deleted genes weighted by their known intolerance to deletions or loss of function mutations, which is the total LOOF score. So, for example, if there was a patient where we saw a 22q deletion in them, that subject would be noted to have a recurrent risk CNV for both schizophrenia and a broader neurodevelopmental disorder risk CNV. And we would also count how many of the genes in that deleted region belong to each of our 18 neurodevelopmental gene sets. along with any other deleted genes that subject might carry at other locations in their genome. Okay, so what did we see? First, as we expected, CNVs previously associated with schizophrenia or broader neurodevelopmental disorders were elevated in our cases. They were found in 1.5 and 2.6% of our cases, respectively, compared to 0.2 and 0.9% of our non-cases. So that was about a 6-fold likelihood of having a psychotic disorder if you had a schizophrenia risk CNV, or a 4-fold increase in schizophrenia risk with a broader NDD-associated CNV. Then looking at our severe phenotypes, starting with child-onset psychosis, 5.6% of the cases with child-onset psychosis had a schizophrenia-associated CNV, compared to 1.2% of later onset cases. And broader NDD-associated CNVs were found in 8.3% of the child onset cases versus 2.1% of the later onset cases. These differences weren't quite significant, although when we looked at schizophrenia and broader NDD risk deletions separately from duplications, we did see a nominal association where having a known NDD-associated deletion was associated with a full-fold significantly increased likelihood of having psychosis onset in childhood. And then in terms of our neurodevelopmental modules and our global CNV burden scores, we didn't see any associations with likelihood of child onset psychosis. However, when we looked at borderline intellectual functioning in our patients, we saw that known schizophrenia and broader NDD risk CNVs were strongly associated with borderline intellectual functioning. So there was a 7-fold increased risk for schizophrenia associated CNVs and a 4.5-fold increased risk for broader NDD associated CNVs. And then when we looked at each of our neurodevelopmental modules, we saw that deletions in the M1 fetal gene regulatory, M11 glia maturation, and M7 protein life cycle modules were associated with borderline intellectual functioning. We also saw some nominal enrichment for the M15 module. And total number of genes deleted was associated, while total LOOF score was nominally associated. However, right, looking at this plot on the left, since there's this really strong association between known risk CNVs and borderline intellectual functioning, and many of those known risk CNVs, they're really large and they delete genes in a lot of these same modules, we really wanted to check, okay, what does this look like after we're excluding the patients who have these established risk CNVs? Like, is there still some kind of biological signal outside of these known risk CNVs? So when we dropped the patients that had known risk CNVs, we saw that M1 deletions showed a similar 2.4-fold increased risk of borderline intellectual functioning that remained nominally associated. And then we realized, given this cognitive functioning phenotype, we could also check if there's a similar effect in non-cases, so in our controls and relatives. And when we did that, we actually did see that M1 deletion showed a similar association with borderline intellectual functioning among the non-cases, and that was also nominally significant. And then if we just pooled the sample across all cases and non-cases, excluding the subjects with known risk CNVs, M1 deletions were indeed significantly associated with likelihood of borderline intellectual functioning, while no other modules or global deletion burden scores showed enrichment. So this is suggesting that M1 deletion burden is associated with poor cognitive functioning across schizophrenia spectrum individuals and also individuals without schizophrenia. And now we also had structural MRI data for about half the cohort, like I mentioned. So one additional question that this raised for us is whether deletions in that M1 fetal gene regulatory module may also be associated with changes in brain structure. And so to get at this, we used the recently released brain charts for the human lifespan data and tool, where in this data set, which characterized normative developmental trajectories of major neuroanatomic features like gray matter volume and cortical thickness in over 100,000 individuals, where with this brain chart, this allows people to then have individual level benchmarking in new data sets for these metrics relative to age and sex matched peers. And these kinds of age and sex normed MRI metrics or centiles were shown to have greater heritability than non-normed neuroanatomic metrics, which is what you can see from the heritability marked by the circles in the plot on the right, compared to the triangles, which are non-normed. So basically these centiles are more closely tied to genetic determinants. So first, as a proof of principle, looking at cortical centile metrics in our cohort where cortical thickness and surface area together are what make up cortical gray matter volume. And cortical thickness, we're just talking about the thickness of the 6 cortical layers, while cortical surface area is the total area covered by cortex for a given region or across the whole brain. When we looked at these metrics across the diagnostic groups with schizophrenia patients in red, relatives in green, and controls in blue, we saw that schizophrenia patients showed reductions in age-normalized gray matter volume, thickness, and surface area compared to controls, which is what we would expect. Relatives were intermediate and also had significantly lower gray matter volume compared to controls, although they didn't actually differ significantly from patients on any of these metrics in our cohort. And now looking at our patients, excluding those with known risk CNVs, because a lot of those known risk CNVs are already known to be associated with either microcephaly or macrocephaly, we saw that M1 gene deletions were associated with a nominally significant increase in gray matter volume. And when we looked at cortical thickness and surface area, it looks like this effect is being driven largely by cortical thickening, which is what is in the middle plot, where a deletion of one M1 gene was associated with a 15% increase in thickness in this cohort. And then when we extended the analyses to the other groups, this effect remained significant with deletion of M1 genes associated with a 13% increase in thickness, whereas the associations with gray matter volume centile and surface area centile weren't significant. Okay, so to recap, we saw that in a cohort of patients with idiopathic schizophrenia spectrum disorders, having a known NDD risk CNV was associated with poor cognitive functioning, and known NDD risk deletions were also found at a nominally significant higher rate in patients with child onset psychosis. And after we accounted for the effects of these known risk CNVs, deletions in M1 genes were also associated with borderline intellectual functioning in schizophrenia spectrum individuals and increased cortical thickness. And we are also seeing these associations when we included controls and relatives. And as I noted previously, genes in this M1 module, they regulate gene expression changes that drive neuronal differentiation in fetal development. And this module is also enriched for risk variants for early onset disorders like autism and intellectual disability. So coming back to this model, if one common path to schizophrenia seems to involve genes that disrupt neuronal excitability and synaptic signaling, that impacts the refinement of synapses and neural circuits during postnatal life and are strongly shaped by common variants with small effect. And the common path to earlier onset neurodevelopmental disorders like autism and intellectual disability includes genetic variants that are disrupting neuronal differentiation and morphogenesis during fetal development, where a greater loading of those risk variants may be due to larger effect de novo mutations or CNVs. The results that we're seeing in this cohort suggest that patients with poorer functioning may have a greater burden overall of damaging variants, such as known risk CNVs for neurodevelopmental disorders defined broadly, and more specifically with damaging variants that affect genes involved in earlier aspects of brain development. And so this is suggesting that using a neurodevelopmental lens to categorize damaging variants may be useful for stratifying patients. Okay, so for the last part of today, I'm going to switch gears a little and talk about how we are trying to use genetics and a developmental framework to move towards prediction of schizophrenia, which is a major focus of ongoing work in my lab. So really, like I was saying at the beginning of this talk, there is still a critical need in psychiatry for us to improve our ability to predict and prevent the onset of schizophrenia and other serious mental illnesses, like bipolar disorder and severe recurrent depression, which are also associated with substantial suffering and disability for many patients. And from a clinical staging framework, we can think about the presence of severe symptoms like full-blown psychosis in schizophrenia or mania in bipolar disorder as representing a relatively late stage of altered brain development and function. And this clinical staging framework also implies that interventions earlier in development, when negative sequelae of severe symptoms haven't yet taken hold and the brain is still more plastic, these kinds of earlier interventions can potentially have a much greater impact on long-term outcome. And support for this kind of clinical staging model for serious mental illnesses comes from evidence that major psychiatric illnesses like schizophrenia or bipolar disorder, they tend to evolve in clarity and severity of symptoms over time. So earlier on, like, somebody who later develops schizophrenia in high school, they might have much more undifferentiated subthreshold psychotic symptoms. We often see anxiety, mood symptoms. So there is this sort of evolution of the emergence of more severe symptoms. And there's also some findings that early interventions can alter disease trajectory for at least some individuals. But if we want to really be able to improve prediction and ideally move towards prevention, we're going to need to be able to better predict SMI outcomes and identify individuals at the highest risk early on during illness progression. But there are challenges in this, right? Like, how do we identify the highest risk individuals early on? How do we determine which early signs and symptoms best predict schizophrenia versus other SMI outcomes, for example? So some possible solutions and general strategies that we are working on in my lab are, one, trying to see if we can identify early antecedents that might be genetically mediated. So basically asking what clinical and neurobehavioral signs and symptoms are associated with genetic risk for schizophrenia, or, for example, for genetic risk for schizophrenia versus genetic risk for other disorders. And we're also thinking about how can we move to build multivariate prediction models in longitudinal data sets where we can incorporate multiple types of data, maybe genetic, clinical, neurobehavioral, to really optimize this. So we are starting to work on this in two complementary data sets. One is the adolescent brain and cognitive development study cohort, where youth were recruited across 21 sites in the US. The total cohort is over 11,000 youth, and it includes over 5,000 European ancestry youth, over 2,000 African ancestry youth, and around 1,500 admixed American ancestry youth. This is a publicly available data set that includes clinical, cognitive, and neuroimaging phenotyping, as well as DNA samples. So really high quality data. It is also a longitudinal study. And so in this data set, in analyses led by one of my PhD students, Mahnor Hayat, what we wanted to look at here is what is the relationship between schizophrenia polygenic risk scores and functioning in childhood. So at the earliest time point we had available in ABCD, when youth were 9 to 10 years old. And specifically what she looked at are functioning in terms of overall cognitive functioning, which was measured from the NIH toolbox, psychopathology symptoms across broad areas of internalizing, externalizing, and other domains using the child behavior checklist, subthreshold psychotic symptoms using the prodromal questionnaire, and lifetime history of any depressive disorder diagnosis, an anxiety disorder diagnosis, conduct disorder, or ADHD based on the Kitty SADS. Okay, and for many people in the audience, this may be obvious, but for folks who aren't as familiar with polygenic risk scores, how did she calculate them? Briefly, to create polygenic risk scores, you start with summary statistics from large genome-wide association studies that tested whether specific alleles someone has at a given spot in the genome. So maybe a spot where people typically have either Cs or Ts, and you're counting basically how many Cs or Ts someone has at a given SNP location. Is that associated with a disorder like schizophrenia? These kinds of GWAS studies, they typically involve tens of thousands of patients and hundreds of thousands of controls now. And then they give you effect size estimates for specific alleles at millions of spots across the genome. And you can then use those to create a weighted sum of the number of risk alleles for the disorder that a new individual in a new cohort carries across the genome. And that's the polygenic risk score for that disorder. And I will just note that one methodological issue that we're dealing with in general as a field is that European ancestry individuals are vastly overrepresented in current GWAS studies. That's what you can see in this plot on the upper right, where European ancestry individuals are in red, right? Even though their representation of the global population is actually a minority in GWAS studies, they are hugely overrepresented. And we also know that the accuracy of polygenic risk scores decreases with increasing genetic distance from whoever was in the original GWAS. So this is just an issue overall for the field because we want to be able to study these relationships in diverse youth. There's some methodological tools. This is something the field is actively working on. So some tools have been recently developed that can help, like PRS-CSX, which take GWAS summary statistics from multiple global populations and try to refine the effect size estimates for alleles. And these kinds of tools do seem to boost the accuracy of polygenic risk scores in non-European ancestry individuals, but we still know that the accuracy of our polygenic risk scores are going to be lower right now in non-European ancestry individuals. Okay, so in the ABCD data set, when McNor tested the relationship between schizophrenia, polygenic risk score, and cognitive and clinical functioning, from the plot on the left, where you have the different dimensional cognitive and clinical outcomes on the y-axis and ancestry-specific analyses on the x-axis, you can see that she saw that among the 9 to 10-year-olds in ABCD, schizophrenia PRS was associated with poorer cognitive functioning across the ancestry groups, and with CBCL-based total, anxious, depressed, aggressive, and rule-breaking behavior in European ancestry youth. She didn't see any associations that were significant for the African-like ancestry or admixed American-like ancestry youth alone in the ABCD cohort for those symptom domains. And then on the plot to the right, looking at lifetime history of diagnoses, even though this cohort is relatively young for having psychiatric disorders, she did see some signal that schizophrenia polygenic risk was also associated with increased likelihood of depressive disorders in admixed American youth. So overall, this was suggesting that schizophrenia polygenic risk is associated with poor cognitive functioning and both internalizing and externalizing symptoms in childhood. Okay, but many youth that have internalizing and externalizing disorders, they don't develop schizophrenia. So just because we are seeing these symptoms or disorders in childhood doesn't mean somebody actually has really elevated polygenic risk for schizophrenia per se, right? And related to that, we also don't really know how specific the relationships are between these clinical and dimensional potential antecedents to schizophrenia. How specific are they to schizophrenia polygenic risk scores versus polygenic risk scores for other disorders or other kinds of risk measures, other genetic or environmental risk measures that we know are associated with psychiatric disorders? And one other thing that I think is also important for us to think about is that if we want to identify genetically mediated antecedents of schizophrenia or other serious mental illnesses so that we can improve early intervention strategies, these kinds of relationships might be the most helpful to look at and be able to identify in individuals that are help seeking or if there's, an elevated risk population, right? If we're thinking about youth clinics, psychiatric clinics, where we want to know among the individuals that are coming in that have ADHD and anxiety symptoms or depressive disorders already, which of them is actually most at risk for developing schizophrenia versus bipolar disorder, for example. Okay, so thinking about what we now know about specific genetic and environmental risk variants associated with different SMI outcomes, we are now exploring these questions. So what early neurobehavioral signs and clinical profiles are associated with genetic and environmental risk for different disorders in an ongoing study that we have with some amazing collaborators at the University of Antioquia in Colombia in South America and at UCLA. So in particular, Juan David Palacio and Carlos Lopez Jaramillo, who are my collaborators at the University of Antioquia, and then Carrie Beard and Luz Oldeluhus and Nelson Freimer at UCLA. We are now working on establishing a cohort of 3,000 PISA youth who are 6 to 15 years at baseline assessment, where this cohort, we are recruiting them to be enriched for risk for serious mental illness defined transdiagnostically. So 1,500 of the youth are being recruited for elevated risk because they have a family member with schizophrenia, bipolar disorder, or depression in prior studies that my collaborators conducted, and 1,200 are being recruited for elevated risk because they have a diagnosis in electronic health records of collaborating hospitals that we know are associated with later SMI. And then 300 youth in the cohort are being recruited to be lower risk because they had a family member in prior studies that who were healthy controls. And then in this cohort, we're getting DNA samples and data on clinical, cognitive, motor, sensory functioning, environmental risk exposures during childhood. And then we're using electronic health records from hospitals in the regions to derive longitudinal clinical outcomes. So like I mentioned, this is still an ongoing study. We've recruited about 1,800 youth so far. And so today I'm going to share some of our really early results that we're seeing in the first 1,100 youth that we've genotyped, who were on average about 10 to 11 years old at assessment. These analyses have been a really incredible joint effort by 3 PhD students in my lab, Mahnur Hayat, Jinhan Zhu, and Anik Shur. Where they've generated numerous psychiatric risk measures, including polygenic risk scores for schizophrenia, bipolar disorder, and major depression, as well as polygenic risk scores for a couple child onset disorders, specifically autism spectrum disorder and ADHD. They've also called copy number variants and identified which of the youth in our cohort have known neurodevelopmental disorder associated risk CNVs. And they've created a cumulative measure of adverse childhood experiences, or ACEs, that we know are strongly associated with risk for SMI outcomes. Specifically here, we're capturing 5 umbrella ACE categories of physical, sexual, emotional abuse, neglect, and bullying. And they've started to look at the relationship between these different risk scores and cognitive, motor, and sensory functioning in the cohort, all of which were measured using the NIH toolbox. The cognitive measure was a cognitive battery across a number of different domains of cognitive functioning. The motor task was a grouped pegboard task, which is basically assessing how quickly somebody can put small pegs into a board with peg holes in a specific order. And then sensory functioning was measured with something called the words and noise task, which is basically assessing how well people can discern specific words over background sort of noise or din of language. And then in terms of clinical outcomes, they looked at intellectual disability and lifetime history of any anxiety disorder, a depressive disorder, conduct disorder, or ADHD based on a Kitty Sads interview. And you can see from the percentages listed here that the rates of psychopathology in this cohort are really quite high, right? So anxiety disorders, even though these youth on average are only 10 to 11, almost 16% of them have an anxiety disorder, almost 22% have met criteria for a depressive disorder, 15% have met criteria for conduct disorder, and about 40% have met criteria for ADHD. And so in terms of associations between the various risk scores in this cohort and early neurobehavioral functioning, here we're looking at our dimensional phenotypes with cognitive, motor, and sensory functioning on the y-axis, and then effect sizes on the x-axis. If there's one asterisk, sorry, the asterisks are so small, that means there's a nominal association in the sample. 2 asterisks means the association survived multiple correction. What we're seeing right now, again, these are very preliminary results, kind of hot off the press. We're seeing that higher schizophrenia PRS in this cohort was associated with poorer sensory functioning. There were actually no associations for bipolar PRS, which is the red dots, or MDD PRS, which is that the brown dots. Higher ADHD PRS was associated with poorer cognitive and sensory functioning. That's the green dots. The presence of a known NDD risk CNV was strongly associated with poorer cognitive functioning. That's the light blue dots. And higher ACE scores, the pink dots, was associated with poorer cognitive and motor functioning. And then in terms of diagnoses, where each of those diagnostic categories are on the y-axis and the odds ratio is on the x-axis, we saw that higher schizophrenia PRS was associated with increased likelihood of conduct disorder and depression, and actually, interestingly, lower likelihood of anxiety disorders. Higher depression PRS was also associated with higher likelihood of depression and conduct disorder. There were no main effect associations for bipolar disorder PRS. ADHD PRS was associated with increased likelihood of ADHD, conduct disorder, and depression. Known NDD risk CNVs were associated with conduct disorder and intellectual disability, and higher ACE scores were associated with all disorders across the board at pretty high odds ratios. And given our relatively wide age range at assessment of 6 to 15, we're also starting to conduct exploratory analyses for interactions between each risk score and age. And we are seeing some interesting signal right now. So we're seeing some nominal interactions for schizophrenia PRS on sensory functioning, where higher schizophrenia PRS is associated with poorer sensory functioning, more so for younger versus older children, which I think probably makes sense if we think about the fact that sensory functioning tends to mature fairly early on. So it may be that we can more detect this relationship among younger youth, but then by the time youth are older, they've sort of caught up in their functioning. And we also are seeing a nominal interaction for bipolar disorder PRS and depression, where the relationship between bipolar PRS and likelihood of a depression diagnosis is stronger at older ages compared to younger ages. Okay, so to recap, where are we? We are seeing that risk variants for schizophrenia seem to converge at the population level on neuronal excitability and synaptic signaling and plasticity pathways, if we're thinking about sort of the average case. It also looks like damaging variants in genes that regulate fetal gene expression changes that might be critical during early neuronal differentiation are associated with poorer cognitive functioning and altered cortical morphology in schizophrenia. We're also seeing that schizophrenia PRS is associated with cognitive, sensory, and internalizing and externalizing symptoms and diagnoses during childhood. But as we've looked at this in our PISA Kids cohort, we're also seeing that so are polygenic risk scores for other child and what we think of as typically adult onset disorders, as well as broad NDD risk CNVs and adverse childhood experiences. And I think it's really important to say that we're also seeing that a lot of psychopathology in childhood seems to be shaped by what are modifiable risk factors, right? ACE scores, elevated ACE scores were really the strongest predictor of likelihood of a psychiatric disorder in childhood. So overall, this is starting to tell us that predicting distinct SMI outcomes really is going to require these multivariate longitudinal models that are incorporating different risk scores and probably a number of different kinds of neurobehavioral characteristics in childhood, so we can really start to understand you know, and get much more precise in prediction for one outcome compared to another. Okay, so with that, there are a lot of people to thank, so many people involved in all of this work that I've shared with you, including the folks in my lab, especially Mahnur Hayat, Anik Shore, and Jinhwan Zhu, who were involved in a lot of the analyses I showed you. The early work that I was talking about in our schizophrenia spectrum cohort with relatives and controls, that was part of a career development award I had from NIH, where my primary mentors, who I owe so much to, Carrie Bearden and Roel Ophoff, as well as my many, many collaborators on that project. And then our Paisa Kids project, Juan David Palacio and Carlos Lopez Jaramillo, Carrie Bearden, Nelson Freimer, and Luz Oldalujus, as well as the many research assistants and staff who are just doing a tremendous, a tremendous job recruiting this cohort, and all of the participants and their families who've been participating. Yeah, I just feel so grateful to work with so many amazing people on this work. And then also our funding bodies, NIH, the Brain and Behavior Research Foundation, and the Scheer Family Foundation. So with that, I will wrap up. And yeah, any questions, I'd love to answer what I can.
Great. Well, thank you very much. And we do, in fact, have some questions that have already been entered. So let's jump right into those. We've got about 4 or 5 minutes, and I want to see if we can get to all of these before we wrap up at 1 o'clock. So the first question came to us early on in your presentation. And the question is that you mentioned heritability genes at 80%. So in our family, there has been only one diagnosis for schizophrenia, but multiple individuals with bipolar disorder or anxiety and panic disorder. So are these all related in terms of genetics?
Yeah, it's a great question, right? And I think that's one that the field is definitely grappling with. And I think that kind of relates to some of what I was saying later on. We know that there's a lot of specific genetic variants that we carry that actually are not associated necessarily specifically with schizophrenia, but might increase risk for multiple kinds of disorders. There can also be shared environmental risk factors. And so, and that kind of set of different psychiatric disorders that people in a given family might meet criteria for at different times. That's a very common clinical picture. And I think it's because there are these risk variants that actually put people at risk for multiple disorders.
Yeah. Great. All right. Another person asks, given that there are environmental risks in schizophrenia, do you have obstetric complications in your schizophrenia cohorts? And if you do have that data, are there gene environmental interactions?
What a fabulous question. So there's a few cohorts that we're talking about here, right? So in the one that we harmonized, where it's that cohort of 1,500 individuals, where about 615 of them have a schizophrenia spectrum diagnosis. Again, that's data where we are harmonizing it across so many studies that my collaborators did over the years, a small subset of whom we have obstetric complication data on. So unfortunately, in that cohort, we've tried to look at it, but I just don't think we have enough data to really look at it. We're still thinking about, is there enough to do something, but I'm not sure that there is. But in our Paisa Kids cohort, we are definitely collecting that. It's retrospective obstetric complication data. So we're using I think it's called the Murray. It's like a classic retrospective scale that often where we're asking about different prenatal and perinatal complications that might have occurred. So we are hoping to look at that longitudinally in that cohort, but we haven't started looking at it yet. But it's a great question because absolutely, I think there could be something there. Yeah.
Okay, great. So it looks like we have one more here, unless somebody else types in a question at the last second. But this final question is, what factors are necessary to define child onset psychosis?
Yeah, it's interesting. So it's not like a DSM diagnosis, right? I think the field has typically called it schizophrenia onset by age 12. I mean, clinically, even these children that have schizophrenia meet criteria for schizophrenia at age 12. Clinically, it's kind of hard, right? And I think this is what we deal with in psychiatry as a field in general, where these disorders have been defined on a surface level, symptom level, right? So it's like they are meeting criteria for classic criteria for schizophrenia, and the field has typically defined that as if you see it by age 12. So it's really just an age thing. In that cohort of 1,500 individuals, when we were looking at childhood onset psychosis, that includes some individuals that didn't meet full criteria for schizophrenia. A number of them did, but there's also, for example, some individuals that might have met criteria for psychosis NOS by age 12. So we were calling that sort of like childhood onset psychosis as we analyzed it.
Yeah. All right, great. Well, it looks like that gets to all of our questions, which is
Okay.
perfect timing then.
Awesome.
Yeah. So thank you, Dr. Forsyth, for being here today. I think it was a great topic. And actually, yeah, I want to thank John Newmeyer for suggesting this topic for one of our grand rounds.
Yeah, absolutely. Thank you so much for having me. And thank you, John, for the invitation. Yeah, it's a pleasure to get to share our work.
Great. And everyone, thanks for joining us. We'll be back again next week. Have a good weekend.